Okay, so my name's Hannah Wilding,
I'm Caroline DeJager.
And I'm Kayla Purcell.
and we did the study of a novel rule for synopsin, simulation of food intake. So for the background of the study, there are a few neuropeptides that relate to hunger and satiety, and one of these is POMC, which is a precursor polypeptide with 241 amino acid residues. AGRP acts to increase appetite and is one of the most potent and longest lasting appetite stimulators, and MPY is a 36 amino acid neuropeptide that acts as a neurotransmitter and influences food intake. All of these are associated with appetite, and they are all neuropeptides. Next, we have the hypothalamus, which is a region of the forebrain below the thalamus that coordinates the activity of the pituitary and controls body temperature, thirst, hunger, and other homeostatic systems. The arcuate nucleus is an aggregation of neurons and nucleus at the bottom of the hypothalamus. The VMH, or the ventromedial nucleus, deals with satiety and prevents the animal from starting to eat. The dorsomedial nucleus deals with satiety as well, and this specifically deals with meal-ending satiety. The lateral hypothalamic nucleus is a collection of neurons that don't really fit elsewhere, but are associated with hunger, so they're placed in this hypothalamus area as well. And the paraventricular hypothalamic nucleus deals with stress and also has neurotransmitters that deal with hunger and satiety.
So Tukey's test is a single-step multi-comparison procedure and statistical test. Ad libitum is a word used for wanted or desired. The ICV bypasses the blood-brain barrier and deposits fluid directly into the cerebrospinal fluid as depicted here. Orexigenic means appetite stimulating. Anorexigenic is loss of appetite. Malaise means a general feeling of discomfort, illness, or uneasiness. And then CFOS is an inducible gene that acts as a transcription factor. It is involved in converting extracellular signals into changes in gene expression and has been linked to cancer when overexpressed. Synopsin is biologically active octopeptide and has antimicrobial capabilities and comes from the African clawed frog. Xenin is a peptide hormone and is also found in humans.
This research was broken up into 8 different experiments. They were each designed in a response to the previous experiment, getting more specific and adding on to the variables tested. So experiment 1 was designed to measure the effect of food and water intake in chicks after a 3-hour fast. The chicks were broken up into 3 groups, all with different amounts of the XPN. Food and water consumption was measured in 30-minute intervals after the injection. Experiment 2 followed the same procedures but measured the food and water intake on chicks that were not fasted prior to injection with lower doses of XPN. Experiment 3 had identical procedures to Experiment 2 but were injected with high doses of XPN. In Experiment 4, chicks either received 0 or 1 nanomoles of XPN and were allowed ad libitum access to food and water. After injection, the chicks were anesthetized and the brains were removed and cut into coronal sections to observe the different nuclei in the hypothalamus and their contribution to CFOS immunoreactivity. Cannulation into the lateral ventricle was utilized in order to set up the 5th experiment. In the 5th experiment, chicks received 3 different doses of XPN 1 millimeter below the cannula previously described. Food and water intake was then measured. Experiment 6 followed the same procedure of XPN injection into the lateral hypothalamus with a crossover design. Experiment 7 measured the mRNA abundance, such as POMC, after the ICV injection of XPN, while Experiment 8 studied the behavior of chicks post-injection.
And so for the results, for Experiment 1, we found that water and food intake were not affected. Experiment 2, food intake increased, but water intake was not influenced, and specifically when there was 0.3 and 0.1 nanomoles of XPN injected, the increase in food intake occurred 30 minutes post-injection. When there was 3 nanomoles of XPN injected, the increase in food intake occurred from 30 to 150 minutes post-injection, and at 180 minutes, the effects were subdued, and there were no differences among the treatment groups. In Experiment 3, there's increased food intake and some decreased water intake. Specifically at 1 and 3 nanomoles injection, there was increased food intake through 180 minutes after injection, and at 9.0 nanomoles, there was a decrease in water intake through 60 minutes post-injection. In Experiment 4, there were differences in CFOS expression in the hypothalamus, and there was specifically an increase of CFOS in the lateral hypothalamus. Those injected with XPN have 150% greater CFOS immunoreactivity compared to those not injected, and there was no effect in the other nuclei of the VMH, DMN, ARC, and the paraventricular nucleus. For experiment 5, the food intake increased after the ICV injection of XPN in the 16-day post-hatch chicks. Specifically, with 0.3 nanomolar injection, the increase in food intake occurred from 30 to 45 minutes post-injection, and with 1.0 nanomolar injection, the increase in food intake occurred at 45 minutes post-injection. In experiment 6, there was increased food intake after the microinjection of XPN in the lateral hypothalamus. Specifically, it was 0.3 nanomole injection of XPN for all chicks, and the increased food intake occurred from 30 to 75 minutes. In experiment 7, there was increased expression of POMC mRNA in the chick hypothalamus with the XPN injected, but there was the abundance of AGRP and MPY mRNA, which are the other neuropeptides, were not affected. In experiment 8, the XPN injected chicks showed different count type behavior and feeding peck patterns, but in the deep rest and deep sitting periods decreased. So for the discussion and the explanation of the results, in experiment 1, the anorexigenic hypothesis was not supported, and the interpretation of the result is that XPN is increasing food intake. So there are no significant results, but that led us to the design of experiment 2, which had chicks non-fasted prior to injection, and that's usually used when an orexigenic effect is expected. In experiment 2, the central injection of XPN stimulates the increase in food intake, and specifically for 3.0 nanomole injection is the most effective dose of XPN central injection for food intake increase. In experiment 3, it is designed to evaluate higher doses, and the lack of food intake stimulation at 9.0 nanomoles shows the dose-response relation is quadratic, and specifically that there was decreased water intake at the 9.0 nanomole XPN injection, and that's likely due to malaise or sickness because of dramatic decrease in water intake. So the central XPN injection is capable of stimulating orexigenic effects only in a narrow dose range. And in experiment 4, the only reactive nucleus is the lateral hypothalamic nucleus, which is consistent with its role in hunger perception. So experiment 5 shows that the dose-response duration varies from the 4-day to the 16-day post-hatch chicks, and specifically the younger chicks, the response persisted to 150 minutes, but in the older chicks, the response of the XPN only persisted to 45 minutes following injection. And additionally, in the older chicks, the threshold of injection for response is much lower. In experiment 6, the receptors associated with the orexigenic response are in the lateral hypothalamus, and the lateral hypothalamus is first order in mediating the XPN response. Experiment 7 showed that it's unclear whether POMC or one of its derivatives is affected, and that the XPN mechanism may include regions stemming from the lateral hypothalamus projections to increase food intake. Additionally, XPN may act alone at the lateral hypothalamus to stimulate hunger, or it may act with confounding with other factors to project through the hypothalamus. And finally, in experiment 8, the XPN has its primary effect on increased food intake, and the other behaviors are secondary.
We concluded that XPN creates an orexigenic effect in chicks, even though the original hypothesis was that the XPN creates an anorexic effect. The response is dependent upon dose, meaning that a high dose may result in malaise, which arises from reduced water intake. The LH and potentially POMC appear to control the orexigenic response. Also, many more factors have been found to cause satiety rather than hunger, which makes XPN unique. This experiment was particularly interesting because XPN has never been studied in this context in terms of the field of hunger and satiety. So one of the things we noticed that was done well was that the experiment followed Virginia Tech's Institutional Animal Care and Use Committee and to the National Research Council publication Guide for Care and Use of Laboratory Animals. One of the things that we did notice is that there hasn't been a lot of research done on this particular topic, and therefore it should be repeated numerous times to increase validity. So for further research, we thought that testing small doses of XPN on larger organisms and potentially people to test the hunger stimulation in anorexic individuals would be an interesting topic. And then also, since xenin suppresses hunger, we could test its impact on people who need to lose weight. Further research on xenin and how it works alongside XPN would be a good topic to discuss because they may cancel each other out or work in a different way when they're combined.
And here are our citations. Thank you very much.